The concepts that we are going to discuss here are energy, chemical reactions, enzymes, metabolic pathways, and the production of ATP through cellular respiration. This is chapter 3. Our objectives for today are to compare and contrast the two classes of energy, describe kinetic energy and chemical energy, understand the laws of thermodynamics, describe chemical reactions and distinguish between reactants and products, describe the general functions of enzymes, describe the pathways that generate ATP, and understand cellular respiration. Energy is the capacity to perform work. It is vital for various cellular processes and biological systems. The two different forms of energy are potential energy and kinetic energy. Potential energy is energy associated with an object's position or stored energy. Examples in cells are observed as concentration gradients across cell membranes. This represents differences in the amount of substances between the inside and outside of a cell. Kinetic energy is energy linked to motion. Energy can be converted between potential and kinetic energy forms. To summarize, potential energy is stored energy or energy based on position, seen in concentration gradients in cells. Kinetic energy is energy in motion. Potential energy can be converted to kinetic energy and vice versa. And energy is essential for performing work and supporting cellular processes. Chemical energy is a type of potential energy. It is stored in chemical bonds of molecules and is crucial in the human body. The key molecules include triglycerides, which are stored in adipose tissue for long-term energy, glucose, which is stored in the liver and muscle tissue, and adenosine triphosphate, also known as ATP, which is present in all cells and is utilized immediately for cellular energy processes. Kinetic energy is broken down into electrical energy, mechanical energy, sound energy, radiant energy, and heat energy. Electrical energy is the movement of charged particles such as electrons along a wire, also known as electricity. Mechanical energy is objects' motion due to an applied force, like a muscle contraction during walking. Sound energy is produced by vibrating objects, causing compression of molecules as in sound waves. Radiant energy is electromagnetic waves traveling through space. And heat energy is associated with random motion of atoms, ions, or molecules and is measured as temperature.
In this video, we will learn about energy. What is energy? Energy is defined as the ability to do work, which is when a force is applied to an object and it moves. Energy is divided into two types, potential energy and kinetic energy. Potential energy is the energy stored in an object. Kinetic energy is the energy that a moving object has. How is energy measured? Energy is measured in joules. Potential energy is the energy an object has because of its position relative to some other object. In a slingshot, the potential energy is stored in the object being flung. Forms of potential energy: chemical energy, elastic energy, nuclear energy, and gravitational energy. Chemical energy. is energy stored in bonds of atoms and molecules. A battery stores chemical potential energy, which is converted into electrical energy. Elastic energy is potential energy stored as a result of deformation of an elastic object, such as the stretching of a spring or a rubber band. Nuclear energy is the potential energy found inside the nucleus of an atom. Gravitational energy is the potential energy of position or place, like a rock on top of a hill and a girl on top of a slide. Kinetic energy is the energy possessed by an object in motion. Forms of kinetic energy: mechanical energy, thermal energy, light energy, electrical energy, and sound energy. Mechanical energy is the energy of an object due to its motion or position. The sum of an object's kinetic energy and potential energy, like a moving car and a windmill. Electrical energy is caused by moving electrical charges called electrons. Thermal energy is energy that comes from a substance whose molecules and atoms are vibrating faster due to a rise in temperature. Light energy. Radiant energy is electromagnetic energy that travels in transverse waves. Sound energy is the movement of energy through a substance in waves. The law of conservation of energy states that energy cannot be created nor destroyed. However, it can be transformed into one form to another. No energy is lost. It is always conserved. Chemical energy from food is converted into mechanical energy. Chemical energy from batteries is converted to light energy. Electrical energy is converted to heat energy. Chemical energy from fuel is converted into mechanical energy. Light energy from the sun is converted to chemical energy through photosynthesis. Mechanical energy from a moving windmill is converted to electrical energy. Electrical energy is then converted to light energy. So again, energy is the ability to do work. It is divided into two types: potential energy and kinetic energy.
Thermodynamics is the study of energy transformation. There are two laws of thermodynamics. The first law is that energy can neither be created nor destroyed. It can only be transformed from one form to another. The second law states that every time energy is transformed, some is converted to heat. Metabolism is the term used for all biochemical reactions in living organisms. Chemical reactions occur when chemical bonds in an existing molecular structure are broken down and new ones are formed to produce a different structure. A chemical equation summarizes the changes in a chemical reaction. The components of a chemical reaction include reactants and products. Reactants are substances present prior to the chemical reaction, and products are substances formed from the reactants in a chemical reaction. So to summarize, metabolism encompasses all biochemical reactions within organisms. Chemical reactions involve breaking down and forming chemical bonds to create new molecular structures. And chemical equations represent reactants and products in a reaction.
You know how sometimes you go to bake a cake, but your bananas have all gone rotten, your utensils have rusted, you trip and pour all of your baking soda into the vinegar jug, and then your oven explodes? My friend, you and your chemical reactions have fallen victim to enthalpy and entropy, and boy are they forces to be reckoned with. Now your reactants are all products. So what are these E words, and what's their big idea? Let's start with enthalpy, an increase or decrease of energy during a chemical reaction. Every molecule has a certain amount of chemical potential energy stored within the bonds between its atoms. Chemicals with more energy are less stable, and thus more likely to react. Let's visualize the energy flow in a reaction, the combustion of hydrogen and oxygen, by playing a round of crazy golf. Our goal is to get a ball, the reactant, up a small rise and down the other much steeper slope. Where the hill goes up, we need to add energy to the ball, and where it goes down, the ball releases energy into its surroundings. The hole represents the product, or result of the reaction. When the reaction period ends, the ball is inside the hole, and we have our product, water. This, like when our oven exploded, is an exothermic reaction, meaning that the chemical's final energy is less than its starting energy, and the difference has been added to the surrounding environment as light and heat. We can also play out the opposite type of reaction, an endothermic reaction, where the final energy is greater than the starting energy. That's what we were trying to achieve by baking our cake. The added heat from the oven would change the chemical structure of the proteins in the eggs and various compounds in the butter. So that's enthalpy. As you might suspect, exothermic reactions are more likely to happen than endothermic ones because they require less energy to occur. But there's another independent factor that can make reactions happen: entropy. Entropy measures a chemical's randomness. Here's an enormous pyramid of golf balls. Its ordered structure means it has low entropy. However, when it collapses, we have chaos everywhere, balls bouncing high and wide, so much so that some even go over the hill. This shift to instability, or higher entropy, can allow reactions to happen. As with the golf balls, in actual chemicals, this transition from structure to disorder gets some reactants past the hump and lets them start a reaction. You can see both enthalpy and entropy at play when you go to light a campfire to cook dinner. Your match adds enough energy to activate the exothermic reaction of combustion, converting the high energy combustible material in the wood to lower energy carbon dioxide and water. Entropy also increases and helps the reaction along because the neat, organized log of wood is now converted into randomly moving water vapor and carbon dioxide. The energy shed by this exothermic reaction powers the endothermic reaction of cooking your dinner. Bon appetit!
Decomposition reactions, also known as catabolism, is when the initial large molecule is broken down into smaller structures. An example is hydrolysis of sucrose into glucose and fructose molecules in the digestive tract. Synthesis reactions, also known as anabolism, is when two or more atoms, ions, or molecules combine to form a larger chemical structure after existing bonds are broken and new bonds are formed. An example is dehydration synthesis reactions during the formation of dipeptide from two amino acids. Exchange reactions are when atoms, molecules, ions, or electrons are exchanged between two chemical structures. An example is the production of ATP in muscle tissue, where phosphate and creatine bond is broken, creatine becomes a free molecule, and phosphate is transferred and bonded to ADP to form ATP. Oxidation-reduction reactions is a type of exchange reaction involving the movement of electrons from one chemical structure to another. Oxidation is molecules, atoms, or ions lose an electron and become oxidized, where they combine or become combined chemically with oxygen. Reduction is a molecule, atom, or ion that gains an electron and becomes reduced. Nicotinamide adenine dinucleotide is a crucial coenzyme found in all living cells and plays a key role in metabolism. NAD+ is composed of two nucleotides joined by their phosphate group. Nicotinamide is one part of NAD+, which is nicotinamide, which contains a nitrogenous base. Adenine, the other part, is adenine, a nitrogenous base also found in DNA. The nicotinamide and adenine parts are connected via their phosphate groups. Nicotinamide is not a component of DNA or RNA. NAD+ is essential for redox reactions in cells, which involve the transfer of electrons. It plays a key role in the production of ATP and is the main energy currency of the cell. NAD+ accepts electrons from another nucleus, becoming reduced to NADH, where NAD+ plus 2 electrons plus 1 hydrogen lead to NADH. The NADH molecule carries electrons to the electron transport chain, where the energy from these electrons is used to produce ATP. The importance of NAD+ is in cellular respiration. It is a vital process. It is vital for processes like glycolysis and the citric acid cycle and oxidative phosphorylation. It's a tongue twister. It is also important in metabolic reactions. It is involved in numerous metabolic pathways, aiding in the conversion of food to energy. So to summarize, NAD+ structure is composed of nicotinamide and adenine nucleotides connected by phosphate groups. They function as an electron carrier, becoming NADH, and is essential for energy production and cellular respiration and other metabolic pathways.
Cells obtain energy during cellular respiration by oxidizing food molecules such as glucose. The energy derived from these oxidation reactions is used to form ATP. Oxidation can be defined as the removal of hydrogens from a molecule. Since a hydrogen consists of a proton and an electron are removed during oxidation. Whenever a molecule is oxidized, hydrogens removed, another molecule must be reduced, hydrogens added to it. During an oxidation-reduction reaction in the cell, an enzyme is involved in transferring the hydrogen, proton plus electron, to a coenzyme called NAD+. This enzyme has a binding site for both the substrate and NAD+. Once the substrate and NAD+ are bound with the enzyme, the hydrogen is transferred from the substrate to the NAD+. In other words, the substrate is oxidized, loses the hydrogen, and the NAD+ is reduced to NADH. As with all enzyme-mediated reactions, once the reaction is complete, the products separate from the enzyme, and the enzyme can be used again. NADH, a high-energy electron carrier, diffuses away and is available to donate the hydrogen to other molecules.
Chemical reaction classification. They are classified based on the relative amounts of chemical energy in reactants and products. Exergonic reactions are defined as the release of energy or energy goes out. Reactants have higher potential energy than products. The example is cellular respiration, where glucose is broken down, releasing energy. Endergonic reactions require energy, or energy is put in. Reactants have lower energy than products. An example is photosynthesis, where energy from sunlight is used to build glucose from carbon dioxide and water. ATP cycling is a continuous process in cells involving the formation and the breakdown of ATP, also known as adenosine triphosphate. This cycle is crucial for supplying energy for various cellular activities. The formation of ATP from ADP and phosphate. The process is the formation of ATP from ADP and an organic phosphate. This is an endergonic reaction, meaning it requires an input of energy. The energy needed for this process often comes from exergonic reactions such as cellular respiration. Splitting of ATP into ADP and an inorganic phosphate. This process is the breakdown of ATP into ADP and an inorganic phosphate. This is an exergonic reaction, meaning it releases energy. The released energy is used to power various cellular activities such as muscle contraction, protein synthesis, and cell division. The ADP and inorganic phosphate produced from ATP breakdown are recycled to form new ATP molecules, maintaining a continuous supply of energy for the cell. The cycle ensures the balance between energy release and energy consumption, which is vital for cells' metabolic processes. ATP is often referred to as the energy currency of the cell because it provides the energy needed for many cellular processes. The ATP cycling process allows cells to efficiently manage and utilize energy.
In this video, we're going to talk about the ATP cycle. So let's begin with a molecule called ADP. ADP is adenosine diphosphate, and it has two phosphate groups. Now we're going to convert this to ATP, adenosine triphosphate, which has three phosphate groups. So to show that visually, here's what we can do. I'm going to put A for adenosine and the two phosphate groups there. And ATP has three phosphate groups. Now, in order to make ATP from ADP, we need an inorganic phosphate. Now, ATP has more energy than ADP. So in order to go from ADP to ATP, we need to put energy in to this process. So because we're putting energy in, this is going to be an endergonic reaction or an endergonic process. Anytime energy is absorbed in a reaction, you have an endergonic reaction. Now let's talk about the other part of the cycle, converting ATP to ADP. In this case, we need to lose a phosphate group. So a phosphate group is going to come out of the cycle. Now, on the first half, or on the left side, we put in energy to make ATP. So in this case, energy is going to come out. The right side is basically the reverse of the left side. So we're going from a molecule that has more energy to a molecule that has less energy. So thus energy will be released. Now because we're releasing energy, that process is known as an exergonic process. Anytime energy is released in a reaction, it's an exergonic reaction. Now there's one more thing that we need, and that is water. To convert ATP to ADP, we need water, and so this is a hydrolysis reaction. We need to use water to break one of the phosphate bonds. Now, if the right side is a hydrolysis reaction, the left side must be a dehydration synthesis reaction. So water is released on the left side. So that is the ATP cycle. That's how ADP converts into ATP and vice versa. Now let's summarize the reaction another way. So when we combine ATP with water, we're going to make ADP, adenosine diphosphate, and we're going to release a phosphate group, and we're going to get energy in the process. So that is an example of the hydrolysis reaction. And as we said before, this is an exergonic reaction since energy is released. Now, on the other side, we have ADP reacting with inorganic phosphate, and we need to put in energy to make this work. And we're going to produce ATP, and we're going to release water as a byproduct. So this process is a dehydration synthesis reaction because we're forming a bond and we're releasing water. We're forming a bond between the two phosphate groups. So because we're putting in energy, or because energy is on the left side of the chemical equation, this is an endergonic reaction. We need energy to drive this reaction forward. And so that's the ATP cycle. In order to make ATP, you need to put energy into it. So ATP is a form of short-term energy storage. But once we convert ATP back into ADP, then that energy that was stored can be released to do useful work, such as powering active transport, or synthesizing molecules, or even driving other endergonic reactions. So ATP is the energy currency of the cell. It's very useful for getting things done and accomplishing many cellular processes. So that's it for this video. Thanks for watching, and don't forget to subscribe.
In irreversible reactions, reactants are converted to products in a manner that results in a net loss of reactants and a net gain of products. These reactions proceed in one direction until the reactants are fully consumed. In reversible reactions, reactants and products are converted back and forth at the rate that maintains equilibrium. This means the forward and reverse reactions occur at the same rate, so the concentrations of reactants and products remain consistent over time. In carbonic acid reactions, carbon dioxide and water combine to form carbonic acid. Carbonic acid is unstable and dissociates into hydrogen ions and bicarbonate ions. This reaction helps in the transport of CO2 from tissues in the lungs where it can be exhaled. The dissociation of carbonic acid into hydrogen and CO3 helps regulate the pH of blood, maintaining acid-base balances in the body. This balance and conversion are vital for maintaining physiological functions and ensuring the proper transport of gases and regulation of pH levels. Reaction rate is the speed at which a chemical reaction occurs. It's measured by how quickly reactants are converted into products. Activation energy is the energy needed to break down existing chemical bonds in reactant molecules. This allows the reaction to proceed. Enzymes are biologically active catalysts that facilitate chemical changes in the human body. They decrease the activation energy required for reactions. Catalysts are substances that accelerate or promote chemical reactions. Enzymes function to accelerate normal physiological activities. The characteristics of an enzyme, they are globular proteins. They range in size from about 60 amino acids to more than 2,500 amino acids. They are produced by protein synthesis within cells. Enzymes can be within the cell. An example is RNA. In RNA, the polymerase, or embedded in plasma membranes, such as lactase, or secreted from the cell, such as amylase released by the pancreas. So to summarize, enzymes are catalysts that reduce activation energy, speeding up physiological reactions. Catalysts are substances that accelerate the reaction. Enzyme characteristics are that they are globular proteins of various sizes synthesized within cells. Enzymes can be found within cells, embedded in membranes, or secreted outside the cell. Cofactors are helper ions or molecules required by enzymes to ensure the reaction occurs. They are non-protein structures. There are 6 major classes of enzymes. Each class has subclasses and different functions. Factors influencing reaction rate include the enzyme or the substrate, which is the substance on which the enzyme acts. The concentration, so reaction rates can be accelerated by increasing enzyme or substrate concentrations. So again, influencing factors can be substrate concentration, temperature, or pH. Metabolic pathways are required for converting a substrate to a final product. They consist of numerous enzymes. Each enzyme catalyzes one progressive change to its specific substrate molecule and then releases the product. Multi-enzyme complexes are groups of enzymes physically attached to each other through non-covalent bonds to form complexes. So to summarize, cofactors are non-protein helpers required by enzymes. There are 6 major classes with specific functions of enzymes. Reaction rates are influenced by substrate concentration, temperature, and pH. Metabolic pathways are a series of enzyme catalyzed steps to convert substrates to a final product. And multi-enzyme complexes are enzymes attached together to form functional complexes.
Is it odd to have a favorite protein? Well, I don't think so. Probably because my favorite protein happens to remind me of one of my favorite childhood games, Pac-Man. If you haven't played Pac-Man before, then chances are we're much, much older than you. But now you can play it on Google. Just Google Pac-Man. It's a Google Doodle. Anyway, I digress. But see, in Pac-Man, you have this little character. It goes around, finds these pebbles, and the pebbles fit right into it. Well, a lot of illustrations that you will find of enzymes, they happen to look, at least us, a lot like Pac-Man. I remember P for Pac-Man and P for protein. Most enzymes are proteins. Now, in the game, we mentioned there are these little pebbles that Pac-Man goes after. Well, enzymes have a specifically shaped area called an active site where items can bind. The items are called substrates. It's very specific binding because the active site is specifically shaped for the substrate that binds there. Very specific. So what happens when a substrate binds an enzyme? Well, usually the substrate is held there with some weak bonds because it's not going to stay there forever. And something called induced fit will happen, which means the active site can change its shape even more to bind that substrate, like an enzyme substrate hug. The enzyme can either build up or break down the substrates that specifically bind to it, and we call the resulting item the product. An enzyme has the ability to really speed up reactions that technically could happen on their own, but with the help of enzymes, they can be sped up to make processes effective for life. Let me give you a great real-life example. The enzyme lactase. Another really cool thing about enzymes is that they often end in ASE, like lactase. Many sugars, on the other hand, end in OSE, and lactose is an example of a sugar. Lactose is a disaccharide, meaning it contains two sugar molecules bound together. We don't actually digest it so well in that form. It's big. The enzyme lactase has the ability to break lactose down into smaller parts that our body can digest. And this is a lot better option than waiting for a chemical reaction with lactose to happen spontaneously. And with the lactase enzyme, lactose can be broken down quickly and digested. Now, there are some people that do not produce enough lactase. They can be what we call lactose intolerant, which means that if they consume foods that have lactose, such as milk, it can make them sick. They can't break the lactose down efficiently without lactase. In that example, one thing to point out, lactase, the enzyme, can break down a lot of lactose, the substrate. The lactase doesn't get used up in the reaction. It's still there. We call enzymes a catalyst because they can be used over and over and over and over in the reaction. By the way, your digestive system uses all kinds of enzymes. You have lipase, and that breaks down lipids, which are fats. You have amylase, which breaks down starch. You have protease, which breaks down proteins. So as you can see, the digestive system is very involved with enzymes. Another thing to point out is that enzymes don't always work alone. Sometimes they get some help. Some often underappreciated but essential little helpers are called cofactors and coenzymes. They may bind to the substrate or to the active site. They help the enzyme do its job of building up or breaking down substrates into products. Now, you didn't forget our Pac-Man analogy yet, right? In the game Pac-Man, there are these ghosts. And when they touch the Pac-Man, it makes this sound. It's like, no, 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 no. And the Pac-Man shape gets all distorted in the process. So what does this have to do with enzymes? No, there aren't ghosts around. But enzymes do have certain ideal conditions that they like. For example, an enzyme that is in your stomach would have an ideal pH that is very acidic because the environment in your stomach is very acidic. Different enzymes have different ideal pH and temperature ranges. If an environment changes out of an enzyme's ideal pH or temperature range, then something that reminds me a lot of that horrible sound I tried to make can happen. The enzyme becomes denatured. That means its shape becomes distorted. It can no longer bind to its substrate. It can no longer work correctly. It is finished. Well, that's a dramatic end to enzymes. Keep in mind that if you have an interest in this topic, many medical researchers have a large focus on enzymes. Enzymes regulate a lot of the body processes, and many diseases can involve specific enzyme production, or the lack of it. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Cellular respiration is a multi-step metabolic pathway. Organic molecules such as glucose, fatty acids, and amino acids are disassembled in a controlled manner by a series of enzymes. Potential energy stored in the molecule's chemical bonds is released during disassembly. The released energy is used to form new bonds between ADP and inorganic phosphate to produce ATP. It is an exergonic or energy releasing process. The organic molecule releases high energy electrons. The energy released from these electrons is used to synthesize ATP. Oxygen is required for ATP production. So to summarize, cellular respiration involves controlled disassembly of organic molecules by enzymes, releasing stored energy. Released energy is used to form ATP from ADP and inorganic phosphate, requiring oxygen and involving high energy electrons. Glucose oxidation is the enzymatic breakdown of glucose within cells. It releases energy to synthesize ATP. It involves at least 20 different enzymes. The enzymes are located in both the cell's cytosol and mitochondria. Cytosol is a semi-fluid content of the cell, and the mitochondria are small organelles within the cell. There are 4 stages of cellular respiration: glycolysis, the intermediate stage, citric acid cycle, and electron transport. So to summarize, glucose oxidation breaks down glucose to release energy for ATP synthesis, requiring enzymes in the cytosol and mitochondria. And the cellular respiration stages are glycolysis, intermediate stage, citric acid stage, and electron transport. Phase 1, imagine glucose, a type of sugar, entering a cell. To start, the cell invests a bit of energy by using 2 ATP molecules. This helps to break down glucose into smaller molecules. As a result, glucose is split into 2 smaller molecules called pyruvate. During this process, some energy is transferred to form 2 ATP molecules and 2 NADH molecules. So in phase 1 of glycolysis, the cell invests a little energy to start breaking down glucose, which eventually results in the formation of smaller molecules called pyruvate, along with a bit of energy in the form of ATP and NADH. After phase 1, we have 2 pyruvate molecules formed from glucose. These pyruvate molecules move into the next part of glycolysis. In the intermediate stage, each pyruvate molecule undergoes a small modification. This modification prepares the molecules to enter the next stage of cellular respiration, the citric acid cycle, which occurs in the mitochondria. No ATP is directly produced during this stage, but some molecules needed for later energy production are generated. So in phase 2, the pyruvate molecules are slightly modified, getting them ready for the next step in cellular respiration, but no additional ATP is directly produced here. The citric acid cycle is also known as Krebs cycle. It takes place in the mitochondria, also known as the powerhouse of the cell. It starts with the 2 pyruvate molecules produced in glycolysis. These pyruvate molecules are converted into a molecule called acetocoenzyme A. Acetocoenzyme A enters the citric acid cycle. During this cycle, acetylcoenzyme A combines with a molecule called oxaloacetate. That's a hard one, to form citrate. Citrate then undergoes a series of chemical reactions, releasing carbon dioxide and transferring electrons to carrier molecules. These electron carriers, such as NADH and FADH2, carry high energy electrons to the next stage of cellular respiration. The citric acid cycle also generates a small amount of ATP directly. So in phase 3, the citric acid cycle breaks down acetocoenzyme A, releasing carbon dioxide and generating high energy molecules like NADH and FADH2, which will be used in electron transport chain to produce more ATP. So here's a summary of glucose digestion. Glycolysis is phase 1, also known as energy investment. Glucose enters the cell. 2 ATP molecules are used to break down the glucose into 2 pyruvate molecules. This process generates a small amount of ATP and NADH. Glycolysis phase 2, also known as the intermediate stage. Pyruvate molecules undergo slight modifications. It prepares the pyruvate for the citric acid cycle, and no ATP is directly produced. The citric acid cycle, also known as Krebs cycle, is phase 3, takes place in the mitochondria. Pyruvate is converted into acetylcoenzyme A. Acetylcoenzyme A enters the citric acid cycle, generating ATP, NADH, and FADH2. Carbon dioxide is released as a byproduct. Electron transport chain and oxidative phosphorylation. High energy electrons carried by NADH and FADH2 are passed along the electron transport chain. The transfer of electrons produces a large amount of ATP through oxidative phosphorylation. Oxygen is required for this process. Water is formed as a byproduct. So in summary, glucose digestion involves glycolysis, which breaks down glucose into pyruvate, followed by the citric acid cycle, which further breaks down pyruvate to generate ATP and electron carriers. These carriers fuel the electron transport chain to produce a large amount of ATP. Electron transfer involves the transfer of electrons from coenzymes NADH and FADH2, which are generated during glycolysis and the citric acid cycle. These electrons are used to form ATP. The majority of chemical energy from glucose is transferred to the coenzymes NADH and FADH2. This energy is released and used to form high energy bonds between ADP and inorganic phosphate, synthesizing ATP. ATP is considered the energy currency of a cell. It provides energy for various cellular activities. Structures involved are located in the inner membrane, also known as the cristae of the mitochondria. The process of forming ATP using the energy from electron transfer is referred to oxidative phosphorylation. So to summarize, glucose breakdown completes with a transfer of electrons to coenzymes NADH and FADH2. The electrons are used to synthesize ATP through oxidative phosphorylation in the mitochondria's inner membrane. Most chemical energy from glucose is transferred to coenzymes and then used to form ATP, the cell's energy currency.
Are you a morning person? One of us is, and one of us is definitely not. Mainly because when I wake up in the morning, it just takes a while for me to get my energy back. It takes a lot of time and coffee for that to happen for me. Cells really don't have that luxury. They are busy performing cell processes all the time. Active transport of many substances needed for their survival, for example. And the energy currency they need, specifically, is ATP. ATP stands for adenosine triphosphate. It's a type of nucleic acid, actually, and it is action-packed with 3 phosphates. We have a video all about ATP and how it works as an energy currency. So where am I going with this? Well, cells have to make this ATP. It doesn't really matter what kind of cell you are. Prokaryote or eukaryote, you have to make ATP. The process for making that ATP can be different, however, depending on that type of cell. One way is called aerobic cellular respiration. Lots of organisms can do aerobic cellular respiration, but this video is specifically going to go into aerobic cellular respiration in eukaryotic cells. That means this video is talking about the process within cells that have membrane-bound organelles, such as a nucleus and mitochondria. Eukaryotic cells include the cells you find in protists, fungi, animals, and plants. The mitochondria, which can be found in most eukaryotic cells, are going to be kind of a big deal in this aerobic cellular respiration because some of the process occurs in the mitochondria. So let's get started. Remember, the major goal for any organism performing this is to make ATP. Okay, here's the overall look at the equation for aerobic cellular respiration. Remember that reactants, inputs, are on the left side of the arrow, and products, outputs, are on the right side of the arrow. This equation, by the way, looks remarkably similar to photosynthesis. Look at how the reactants and products are on different sides. While that doesn't mean they're simply opposites, it does show that they have substances in common. In photosynthesis, organisms make glucose. Notice how glucose is a product. But in cellular respiration, organisms break the glucose down to make ATP. Fun fact, you know how when a bean seed first germinates in the ground, it isn't able to do photosynthesis yet? Yeah, the germinating bean is relying on glucose that it has stored, and it's doing cellular respiration to break it down to make ATP so it can grow. Of course, once it starts to mature and develop leaves, it can do photosynthesis. It will be able to do both photosynthesis and cellular respiration. Plants make glucose in photosynthesis, and they break it down in cellular respiration. They can do both. But if you aren't photosynthetic, such as a human or an amoeba, you have to find a food source to get your glucose. You need glucose to get this process started, and we're going to assume that we're starting with one glucose molecule. Step 1, glycolysis. This step takes place in the cytoplasm, and this step does not require oxygen. It's considered anaerobic. In glycolysis, glucose, the sugar from the equation, is converted into a more usable form called pyruvate. Glycolysis usually takes a little ATP itself to start up. The net yield from this step is approximately 2 pyruvate and 2 ATP molecules. And 2 NADH. What is NADH? NADH is a coenzyme, and it has the ability to transfer electrons, which will be very useful in making even more ATP later on. We'll get to that in a minute. Now an intermediate step occurs. The 2 pyruvate are transported by active transport into the mitochondria, specifically the mitochondrial matrix. In the mitochondria, pyruvate is oxidized. The 2 pyruvate are converted to 2-acetyl-CoA, which will be used by the next step. Carbon dioxide is released and 2 NADH are produced. Step 2, the Krebs cycle, also called the citric acid cycle, still in the mitochondrial matrix. The citric acid cycle is considered an aerobic process. While the cycle doesn't directly consume oxygen, some of the events in the cycle need oxygen to continue. To learn more about this intricate cycle where the 2-acetyl-CoA will enter, check out the further reading suggestions in the video details. Carbon dioxide is released. We also produce 2 ATP, 6 NADH, and 2 FADH. FADH is also a coenzyme, like NADH, and it will also assist in transferring electrons to make even more ATP. Step 3, the electron transport chain in chemiosmosis. This is just a beautiful thing, really. In eukaryotic cells, this is still in the mitochondria, and to be more specific, it's involving the inner mitochondrial membrane. We do require oxygen for this aerobic step. This is a very complex process, and we are greatly simplifying it by saying that electrons are transferred from the NADH and FADH to protein complexes in electron carriers. The electrons are used to generate a proton gradient, as protons are pumped across to the intermembrane space. All these protons being pumped out into this intermembrane space generates an electrical and chemical gradient. The thing is, if you remember from our cell transport video, ions, like the H+, don't easily travel across membranes directly without something to travel through. The protons can travel through an amazing enzyme called ATP synthase. If I could be any enzyme, I'd be ATP synthase because it has the ability to make ATP by adding a phosphate to ADP. ADP is a precursor to ATP. ADP has 2 phosphates, but if it obtains a 3rd phosphate, it becomes ATP. So, in chemiosmosis, the protons travel down their electrochemical gradient through a portion of the ATP synthase, powering it to make ATP, the ultimate goal. Oxygen is the final acceptor of the electrons. When oxygen combines with two hydrogens, you get H2O, water. If you remember from our equation, water is a listed product. Now, the electron transport chain in chemiosmosis step makes a lot more ATP compared to the other two previous steps. How much? So I've noticed in my years of teaching and the assortment of textbooks I've collected over the years, there are some different numbers in cellular respiration charts. In fact, you can even see them change a bit in different editions of the same book. And it's made me want to emphasize that it's important to not just memorize a number, because it really is more of a range. I want to focus less on the number of ATP made per glucose molecule because it depends on a lot of variables. One variable is the gradient we were talking about. How many protons were pumped across the mitochondrial membrane? You can see more variables discussed in the factual references, and those references have estimates ranging from 26 to 34 molecules of ATP per glucose molecule in the electron transport chain in chemiosmosis step alone. Then, if you add the other two steps, Krebs, otherwise known as the citric acid cycle, and glycolysis, you could estimate a range of anywhere between 30 to 38 net ATP molecules total per molecule of glucose. Again, to emphasize a range. Now, this was just one way of creating ATP, but like we had said at the beginning, all cells have to make ATP, but the way they do it can differ. If there's no oxygen available, some cells have the ability to perform a process known as fermentation. It's not as efficient, but it can still make ATP when there isn't oxygen. We have a video about that. We can't emphasize enough how important the process of making ATP is for cells. For example, cyanide, which is found in some rat poisons, can block a step in the electron transport chain, which would block ATP production. A poison that prevents ATP from being produced can be deadly. With the important role that mitochondria have in ATP production, there is also a demand for increased research on mitochondrial diseases. We are confident that the understanding of how to treat these diseases will continue to improve as more people like you ask questions. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So to summarize the stages of cellular respiration, the first stage is glycolysis. Its location occurs within the cytosol. It is the process of the breakdown of glucose into pyruvate. There's a net production or a net gain of 2 ATP molecules. In the intermediate stage, also the second phase, it occurs within the mitochondrial matrix. The process is the conversion of pyruvate to acetocoenzyme A. 2 CO2 molecules are released per pyruvate. The third phase is the citric acid cycle or Krebs cycle. The location is within the mitochondrial matrix. And the process is a series of enzyme catalyzed reactions. 4 CO2 molecules are released per glucose molecule. 1 ATP molecule is formed per turn. The fourth phase, or the oxidative phosphorylation, occurs within the inner mitochondrial membrane, also known as the cristae. Electron transport chain and ATP synthesis activity occurs. 30 ATP molecules are generated through oxidative phosphorylation, including the electron transport chain. So the total production of ATP in glycolysis, there's 2 ATP, the citric acid cycle, 2 ATP from 2 turns in the oxidative phosphorylation. There's 30 ATP with a total of about 34 ATP per glucose molecule. And that is the end of the lecture.